So exocytosis, endocytosis. So this is a very primary transporting mechanism. When you look at this cell, so this is the cell, basic structure of the cell, nucleus. So cell has got different transporting mechanism, oxygen, carbon dioxide, transports. So these elements which essentially transports across the cell membrane with simple diffusion process, whereas we have some more elements, electrolytes. Those are sodium, potassium, chloride, calcium, magnesium, etc. So these potentials, electrically charged potential, also crosses the cell membrane, across the cell membrane with these specialized gated channels. Okay, and then we have got water transportation across the cell membrane also through aquaporins. Aquaporins are the specialized the protein channels which allows the water passes across the cell membrane. And now we have got transportation mechanism of exocytosis and endocytosis. So exo and endocytosis are the primary transporting mechanisms. Primary transporting mechanism are very essential primary transporting mechanism. Essential primary transporting mechanism. Then what does it transports? Unlike these elements, so these exo and endocytosis transports the larger molecules and bulk quantity. Larger molecule, bulk quantity transport usually takes place with the help of exo and endocytosis. Okay, so in this case, exo and endocytosis, the material essentially transports across the cell membrane by the help of the cell membrane vesicle itself. The cell membrane pinches off and becomes a vesicle. Okay, and the cell membrane forms a transporter, cargo. Cell membrane itself forms a cargo, and then this cargo go into the further interior parts of the cell. And then, so at the same time, similarly, the exocytosis is also simply a cargo, the loaded cargo, the loaded vehicle, loaded with material, substances, is essentially initiating from the Golgi apparatus, and then it eventually reaches to the cell membrane and expels its content, okay, contents into the interstitial fluid. This is how exactly exocytosis happens. So now, despite of having all transporting mechanisms, so this exo and endocytosis process are the primary transporting mechanism which transports the large size molecules in bulk quantity. This is the essence. And now, coming to the exocytosis, where does it happen? Okay, supposedly when you look at the cell, so endocrine secreting cell, I just wanted to look at, the insulin secreting beta cells. We have different Golgi apparatus. So it pinches off one vesicle from the Golgi apparatus, starts, and then this vesicle gradually reaches to the peripheral part, and then this vesicle releases its contents, so into the interstitial fluid. This is how the exocytosis process happens. So the vesicle loaded with the contents. This is the, in case of insulin, so this is the beta cell. In case of insulin secretion, this is the insulin tertiary processed insulin. The protein insulin is eventually reaching to the interstitial fluid via the cargoes, loaded cargo, right? So vesicles are releasing the contents. And when you look at the neuronal cells, neurons, so neurons has got communication with the synapse, and we have number of vesicles, okay, filled with the neurotransmitters. These neurotransmitter, whenever there is an action, potential hits the terminal of the presynaptic part of the neuron, it releases the the chemical neurotransmitter into the cleft, and then thereby it binds to the postsynaptic cleft. So this is how endocytosis happens in the neurons. Sorry, the exocytosis happens in the neurons. So examples of the exocytosis, very simple example, is secretion of the hormones through the hormonal glands, and secretion of exocytosis, part of the neurotransmitter in the neuronal system, central and peripheral neuronal system. And then how fast the exocytosis can happen, how fast. So I just went on reading some articles. The maximum speed of neuron, maximum speed of action potential, neuronal transmission touches up to 130 meters per second. Per second, 130 meters is the maximum speed of neuronal action potential transmission. So in this case, what happens? The neurotransmitters also has to release very fast manner in order to cope up with the speed of transmission of the neurotransmission. Communication, according to communication transfer, the speed of the transfer, the neurotransmitter also has to release from the presynaptic to the postsynaptic cleft. That means the exocytosis process happens in very fast manner, particularly in the central nervous system. Okay. Now coming to the point of cargo transfer from here to the here. Cargo is forming somewhere the stacks trans area of the Golgi apparatus, stacks of the Golgi apparatus. From there, eventually reaching to the peripheral part of the cell, and then thereby releasing. There are designated transporters. So remember, the exo and endocytosis process exclusively depends on ATP. Exclusively depends on ATP at various levels. Use ATP essentially utilizes in the process of exo and endocytosis. Here, so once the final form of hormone is formed, so hormone is packed with the vesicle, and this vesicle has to transport from here to here. So there is a transporting mechanisms. So in order to transport this vesicle, formed vesicle, from here to here, we have certain transporting mechanism in the cell, interior cell. So we have some scaffolding proteins in the cell, scaffolding structures, protein structures in the cell. Those are called microtubules, microtubules and microfilaments. Microtubules, these are the microtubule and microfilament, for example. So these are the scaffolding structures, scaffolding protein structures present in the cell, which provides the basic structure, shape or structure of the cell. And these microtubules and microfilaments acting as a roots, ways to transport these vesicle from one area to the other area, designated area. How does it transport? Exactly before starting its journey, any vesicle, it is pre-decided where it has to reach by certain physiological process. And there are three families of molecular motors. One is dynein, kinesin and myosin. Dynein, kinesin and myosin, these three fellows are the molecular motors. One fellow here, and then one more fellow. So here, simply I just drawn the schematic diagram. One fellow is moving in direction, one fellow this direction, one fellow this direction. So this is called dynein, kinesin, myosin. So this heads is called vesicle. These heads are vesicle. So cargo filled with the substance. It can be protein or it can be any substance, right? So they move on towards designated area. So pre-decided destination is fixed here. Once before it starts journey, these vesicles are transported with these molecular motors, dynein, kinesin and myosin. So with the help of the microtubule and microfilaments, which are scaffolding proteins in the cellular environment. So for example, this is the microtubule. Okay, so it can be kinesin, dynein and myosin. Okay, myosin of course work on the actin filaments. Kinesin, dynein, for example, though this is a round structured vesicle, so these kinesin working on the microtubule like this. Dynein works microtubule on this reverse direction. Always kinesin in one particular direction, kinesin always moves towards the positive end of the microtubule, and whereas dynein always moves towards the negative end of the microtubule, because microtubule also in polar nature. Simply they walk on simply like this. So it transporting the vehicles. It transports vehicles. These are the transports vehicles. So these three superfamily molecular motors can be seen in the exocytosis and endocytosis process. In both process, any transporting, any transport vehicle has to transport it along with the help of these molecular motors, right? And again, this exocytosis process, usually some books mention these are regulated. Some exocytosis process is regulated, happens through the regulated pathway. Some exocytosis process, some vesicles are unregulated, partially regulated, usually partially regulated. All are regulated pathway. Highly processed protein usually transfers to the interstitial fluid. So regulated, unregulated pathway. So there is no much difference. Regulated pathway is the pathway with highly processed molecules or substances are transporting, whereas unregulated pathway, partially processed proteins are transported. That's all the difference between both, right? So this is about the exocytosis. And coming to the endocytosis. Endocytosis, we have three varieties. One is pinocytosis, means cell drinking. Second one is receptor mediated endocytosis. Receptor mediated endocytosis. And thirdly, the phagocytosis. Phagocytosis. Let's see the pinocytosis. What is pinocytosis? So we have, we have brush border epithelial cells on gastrointestinal tract. So they have to engulf, they have to uptake all the materials or substance, whatever being digested at the digestive lumen. So they have to uptake. So here the brush border epithelial cells mostly perform the pinocytosis process to uptake the nutrients. Nutrients, those are soluble vitamins, soluble fats and other elements, essential elements, micro elements. Usually pickups by these brush border epithelial cells in the gastrointestinal tract from the lumen. The absorption process, usually the absorption process takes place with the help of pinocytosis. Pino means cell drinking. So majority in this process, majority of the material are with different sizes. It extends simply the pseudopodia and then it engulfs the area where majority volume contributes with the fluid. That's why the process named after pinocytosis. The fluid contribution is more. The composition, whatever the vesicle is formed, so water or fluid is filled with this vesicle, majority part, and certain extent of large molecules and some of small molecules, variable molecules are there, substances, right? Soluble vitamins, soluble fats and essential elements. These are the main contents. So this is called non-specific endocytosis. Non-specific, it is not a specific, means it is engulfing both fluid, small molecule and large molecules. Mixture, right? So the similar process, this is called primary endosome, primary endosome. So gradually it becomes late endosome, late endosome, and gradually it becomes, and then it mixes with the lysosome. So lysosome is coming somewhere. This is a lysosomal mixer. This is called lysosome. Lysoendosome. Okay, lysosome, where the contents are digested, degrade or destroyed. Digested, degrade or destroyed. So similar process. Transporting mechanism also just like exocytosis process. So the vesicle transport is also takes place with the help of dynein, kinesin and myosin on simply walking on microtubules and microfilaments. Okay, so this is about the pinocytosis. Pinocytosis usually seen in the mesh border epithelial cells of the gastrointestinal tract for the absorption of the digestive material, which is mostly along with the fluid. And coming to the receptor mediated endocytosis. What is the receptor mediated endocytosis? Again, this is the cell. So we'll find many pits usually in the cell on the cell membrane. There are many pits. Okay, so somewhere around in the pits we have receptors for particular ligand substance to absorb. So before going into the details, I would like to mention where does receptor mediated endocytosis happens. Where does receptor mediated endocytosis happens? For example, LDL receptors, uptake of the cholesterols by the cells is happening through the happening through the receptor mediated endocytosis, cholesterol uptake, and iron uptake, transferrin receptor, iron uptake, transferrin receptor, TFR, also happening to the receptor mediated endocytosis. Growth substances, certain growth substances also go through this receptor mediated endocytosis, and certain hormones and certain substances, particularly some specific hormones also go through this kind of mechanism. Endocytosis, of course, it is a part of endocytosis, but receptor mediated endocytosis. Why? Because receptor mediated endocytosis is specifically designed for the one specific ligand. For example, it may be designed for the iron molecule because it may be transferrin receptor. Only iron can bind here and only iron can go inside, unlike pinocytosis. This is the difference. The pinocytosis, anything which is there in the vicinity of the endocytosis will just trapped by the cell membrane, right? And then receptors. Just below the receptor, we have one special protein called adapter protein, adapter protein. Just below that adapter protein, we have very special protein called clathrin. Clathrin protein is a very essential protein which is just arranged like a lining, bricks lining below the pit of the cell membrane. So where it has got attachment with the adapter protein and thereby receptors, specific receptor. It can be transferrin receptor, it can be LDL receptor, it can be receptor for some growth factors, or receptor for some specific hormone, anything. Once the ligand binds, once this target is bind with the receptors, right? Once the target is bind with the receptor, what happens? So it forms the gradual vesicle. It forms a gradual vesicle. Vesicle forms. So vesicle, around the vesicle we have clathrin, surrounded. Clathrin protein is surrounded, and then so after that what happens? It releases from the cell membrane, forms the vesicle, and here we have what is a receptor also, receptor, something like that, and again clathrin. So this is how the endosome is formed. The clathrin plays a very important role here. Clathrin is acting as a initiator of the vesicle and formation of the vesicle and stabilization and releasing of the vesicle from the cell membrane. Snipping off, budding off process also initiated by the clathrin. So this is the main functions of the clathrin in the receptor mediated endocytosis. Then how does the clathrin looks like? Clathrin, simply I'm just drawing here. So this is a central part of the clathrin, has got 3 arms. These are clathrin triskelion. Tri means 3. There are 3 arms for the clathrin, and so 3 arms, attached with the one arm is attaching with the other arm. So automatically it forms a round lattice. The lattice of the arrangement, the geometric arrangement of the clathrin protein looks such a way, it looks exactly like a ball. So I got a ball, children playing ball from my home, right? So it exactly gives the shape of this ball, this vesicle, right? Exactly. So geometric arrangement of this clathrin, this is the center part of the clathrin. This is one arm, this is another arm, this is another arm. So there are 3 arms, right? And again here, this is the center, 3 arms here, this is the center, 3 arms here. So something like it exactly forms the hexagonal, geometric arrangement. So across the vesicle. So it forms the complete vesicle. It looks exactly like this, the geometric array of this triskelion interlocking by the clathrin protein. So once it completely forms the vesicle, the clathrin will release from here. And automatically reaches back to the cell membrane by the help of heat shock protein by the hydrolysis of AP, adaptor protein. This green one, hydrolysis, initiates the releasing of the clathrin from this ball, and this becomes one endosome. Then endosome move on its own to the destinated area by the help of microtubule, microfilaments, and 3 superfamilies of molecular motors, that is called dynein, kinesin, and myosin. So where finally the contents may be released or digested and may be destroyed in the cell. So because as the endosome moves, primary endosome, late endosome, lysosome, so what happens? The pH also decreases. It becomes more and more pH, more and more acidic as the endosome moves towards the destinated area. Okay, and one more thing I would like to mention here, how exactly this endocytosis, this endosome moves from one place to the right destinated place. So this happens like exactly the airport baggage sorting system, luggage sorting system. When I drop in my baggage at the airport, the particular baggage, my baggage, will reach to my destinated flight. That's how exactly the airport, the baggage system will sort to reach the right destination on right time, right? So this is how exactly the RFIDs will maybe tag to these vesicles, just like RFIDs, radio frequency identification. So we can track everywhere where it is going and where pre-decided. So there is a code attached to the there is a code given to this vesicle, and particular code is tagged to reach particular destination. Then gradually, like a baggage system in the airport, this vesicle from one place to the designated place reaches exactly. So that usually happens in all the processes. Exocytosis or endocytosis are these sub processes. Okay, and then this is very interesting point. Reaching to the very particular destination. Reaching to the very particular destination. Radio frequency ID, RFID, radio frequency tagging, something like that. Something technology is there. Something similar technology may be there in the cell internal environment. So exactly the vesicle is reaching to the particular designated area. So lastly, phagocytosis. Coming to the phagocytosis. So far we have discussed these things. Phagocytosis means eating the cell. Phagocytosis. Phago means eating up. Where do we see this phagocytosis? Essentially we see the professional killers, first line of defense in our body, WBCs, mainly neutrophils and monocytes, and even reticular endothelial cells, those are called macrophages. These three cells are very professional and involved in the phagocytosis. Mainly they may engulf the foreign body, living organism, they may be engulf the dead cells, they may be engulfing the debris, and the clearance of the debris and all happens through this phagocytosis. When you look at the primitive organism, that is called amoeba. So mainly amoeba also simply depends on food material. This process is called, this is some organism, food organism. It usually extends the pseudopodia here and here, and then gradually engulf this the organism, and then it go on. The phagosome go on and then digest, and after that it will release the remaining, expel the remaining contents, digested contents. So this is called phagosome. In case of pinocytosis and receptor mediated endocytosis, this was called endosome, whereas in phagocytosis this is called phagosome. And early phagosome, late phagosome, lysophagosome, and then expel the contents in the amoeba. This is how the basic, the primitive cell, the basic food process, food intake process primarily happens occurring through the phagocytosis, right? So this is what exactly the endoexocytosis process takes place in the living organism, in the cells.